evening to one and all present here. Today we are going to discuss about the intermediary metabolism, first unit 1. So today we are going to discuss about the what is metabolism and glycolysis pathway. So already we discussed what is metabolism means. Metabolism refers to the series of chemical reactions it occur in the living organism to sustain life. So what we are taking the food in through to the diet, the chemical energy, that is the macromolecules, carbohydrate, fat and others, proteins, amino acids, others. It is converted to the chemical waste, carbon dioxide and water, and the energy currency of our body is to requirement of the ATP. There are two types of our metabolic process. One is catabolism and another one is anabolism. Catabolism means that is breakdown of the molecules and anabolism means synthesis of the molecules. In the catabolic process, that is the transfer of the energy, the complex molecules to ATP, it is converted to the simple molecules of glucose, amino acid, glycerol and fatty acids. So the anabolic process, the transfer of energy from the complex molecules, that is the complex molecules such as starch, protein and lipids. So the catabolism, both catabolism and anabolism, the metabolism is mainly to required for the nutrition and the existence, that is the nutrition's requirement. The requirement is in the form of ATP. So the bioenergetics, it is described the metabolism of the biochemical pathway in which it is cells obtained from the energy. So one of the major aspects to form of the energy, to forming of energy. So this metabolism, it is otherwise it is called as a energy metabolism. So we require the nutrition of the energy, the nutrition that get produces necessary to synthesize for nucleic acid, proteins and other biomolecules in our body. So this nutritions, the various substances, our body requirements which are either insufficient or insufficient, resulting in poor health conserving metabolism. So necessary nutrients, it is help to supplying for the required of energy to other chemicals that our body cannot be synthesized of its own. So through to the food only we are going to be take for the diet. So this diet requires both, it occurs both organic nutrients and inorganic nutrients also. So organic nutrients, it includes the vitamins, carbohydrate, proteins, etc. Then inorganic chemical compounds, oxygen, water, metals and etc. So the carbohydrate metabolism and protein metabolism and lipid metabolism. In first unit, we are going to be discuss about what is carbohydrate metabolism. In that carbohydrate metabolism, we are going to discuss about the what is glycolysis pathway. Okay, so carbohydrates are supplied in the three forms. One is starch, sugar and cellulose. Then the protein in metabolism, the proteins are very important for to building block of our cells. Already we know. So these proteins, some of the proteins or amino acids cannot be synthesized in our body. So through to the food only we are going to be take. So like lysine, tryptophan, methionine, isoleucine, leucine, valine, threonine. So these are all the examples of the some amino acids. Then, so how to increase our metabolism? So first we discuss what is the definition of metabolism and what are all the types of metabolism and how we are going to be increase our metabolism. So to be fit for our healthy, we need to avoid for more calories to intake. So we eat to deliver the energy our body, it perform its function. So we have to eating too little quantities could slow down our metabolism. Then, and body cannot provide the essential nutrients as per the research leads to weight loss, which is muscle mass and not fat mass. So having proper breakfast is boost up our metabolism. So skipping our morning breakfast are more likely to poor our metabolism. So we don't skip our breakfast in morning because it lower the energy level of our metabolism. So the caffeine, it stimulates our central nervous system to activate our metabolism rate by 5 to 8%. So according to researchers, the more fiber intake, it is burning the fat about 30%. Then it include more fiber in their diet. So we have to remain healthy. Healthy in the more fiber we have to take means we have to fit and healthy. So include more organic foods and fruits also we have to take to increase our metabolism. So next we move on to what is a basic metabolic pathway. So glucose, already we know what are the key components that enter in the metabolism. It is already we are all well known about that is fatty acids, glucose, glycerol and amino acids. So all these metabolic pathways, what is the requirement? The end product is ATP, adenine triphosphate. So glucose is converted to pyruvate and acetyl-CoA. Acetyl-CoA is a precursor of TCA cycle, otherwise it is called as a Krebs cycle, and finally electron transport chain. So this is the basic metabolic pathway in our body. So what is anabolic? There are already we discussed what is anabolic pathway, catabolic pathway and ambibolic pathway. Anabolic pathway means the chemical reaction, the larger molecules it should be synthesized. Anabolic. Catabolism means breakdown of the molecules. So then ambibolic means the pathway of both anabolic process and catabolic process, it should be occur. It is called as ambibolic pathway. So the intermediate or metabolic and catabolic pathway can also act as a precursor for any anabolic pathway, example TCA cycle. So after glycolysis we are going to discuss about what is Krebs cycle or TCA cycle. Then we have to definitely know what is the differentiate between anabolism and catabolism. One is building up process, here breakdown process. Then anabolism is smaller the molecules, the catabolism is larger molecules break into smaller molecules. Chemical energy is formed and stored in anabolism, and catabolism, the stored chemical energy is released and used. Here anabolism example is synthesis of protein from amino acids. In catabolism, that is the breakdown of glucose to carbon dioxide and water. So this is the mainly differentiates between the catabolism and anabolism. So what are the fate observed of carbohydrate? Already I told that is the fate of carbohydrate means the glucose metabolism, it should be involved in the multiple process. That is glycolysis, glycogenolysis, gluconeogenesis, glycogenolysis. So these are all the pathways which involving in the multiple process of glucose metabolism. So the glycolysis is mainly, we know that is liver is the major organ. So all our metabolic pathway should be occur in the liver. So in glycolysis, the liver is the process. It involves in the enzymes to encourage for the glucose catabolism in the cells. So the digested molecules of food, what we are taking in the diet, it should be absorbed in the small intestine and absorbed in the bloodstream to transported through to the bloodstream, it should be transported to the liver. So the final product of carbohydrate digestion in monosaccharides such as glucose, galactose and fructose, etc. So these are all the products of the final product of the carbohydrate digestion. Whatever we are taking, whatever we take for our diet, the final product it should be that is in the carbohydrate, that is the galactose, fructose and glucose are the final product. Final product of the digestion. The individual monosaccharides are absorbed into the bloodstream and transported to the liver. The final product of carbohydrate digestion in monosaccharides are glucose, galactose, fructose and etc. The molecules it should be absorbed through to the bloodstream and transported into the liver. So this is the fate of the carbohydrates. Then we move on to what are all the characteristics of the metabolism. First, we already discussed what is the definition of metabolism and what are all the types of metabolism and what are all the difference between the catabolism and anabolism and what is the fate of carbohydrate metabolism. Then we move on to what are all the characteristics involved in the metabolism. So first one is the metabolic pathway are irreversible. Okay. The second one is every metabolic pathway has committed first step. all metabolic pathways are regulated. Then metabolic pathways in eukaryotic cells, it occurs in the specific cellular locations. So first today we are going to discuss about the glycolysis pathway. So glycolysis means it is a Greek word. So glycolysis means it is a Greek word. Glyx means it is a sweet. Glyco means it is a sweet. And lysis means it is breakdown or splitting. So this glycolysis pathway, otherwise it is called as a EMP pathway, that is Embden-Meyerhof-Parnas pathway or EMP pathway. So next, the introduction of the glycolysis or definition of the glycolysis pathway. So totally in glycolysis, there are totally 10 enzymes involved in that to catalyze the reaction to conversion of glucose to the pyruvate. Okay, totally 10 enzymes involved. Already we know the chemical reaction that should be occur in our metabolism is, so that is the coenzymes and cofactors, both are very important. So in this oxidative process, 1 mole of glucose is partially oxidized to 2 moles of pyruvate. So the major pathway of glucose metabolism, it occurs in the cytosol of our cells. The unique pathway, it should be occurs in aerobically as well as the anaerobically, and it does not involve in the molecular action. So two types of reactions, a pathway it should be occur, that is both anaerobic pathway and aerobic pathway. Presence of oxygen and absence of oxygen. Then it is also include to form of the lactate and pyruvate. Goes to pyruvate and two products, that is pyruvate and lactate, aerobic and anaerobic. So the glycolytic sequences of reactions differ from species to species. Only in the mechanism of its regulation, it is subsequent metabolic fate of pyruvate form. So in last, we are going to discuss about what is the fate of pyruvate involved. So before that, we have to discuss what are all the enzymes involved in the glycolysis pathway, how the glucose it should be converted to the pyruvate. So the in aerobic organism, the glycolysis that is prelude for the citric acid cycle and ETC. ETC means electron transport chain. The glycolysis is the central pathway of the glucose catabolism. So there are 2 phases occur in glycolysis. Phase 1 is preparatory phase and phase 2 is payoff phase. The glycolysis, it leads to the breakdown of 6-carbon glucose into molecules of 3-carbon pyruvate with the enzyme catalyzed to categorized into the 2 phases, that is preparatory phase and payoff phase. That is the 6-carbon molecules, it is converted to the 3-carbon molecules. So it occurs in the totally 10 enzymes involved in the metabolic pathway, and also it categorized into the 2 phases. One is preparatory phase and phase 2 is payoff phase. So first we are going to discuss about the preparatory phase. Only first to 5th step. First 5 steps are involved in the glycolysis. The glucose is, which it is enzymatically phosphorylated by ATP to yield fructose 1,6 bisphosphate. This fructose 1,6 bisphosphate, in the presence of enzyme aldolase, it split into 2 products, that is glyceraldehyde 3-phosphate and DHAP, that is dihydroxyacetone phosphate. So up to this, it is called as a preparatory phase. So listen please carefully, because in glycolysis there are 2 important categories of the phases are involved. One is preparatory phase and payoff phase. In preparatory phase, first 5 steps involved in the preparatory phase. The next 6 to 10 steps are involved in the payoff phase. In preparatory phase, it consists of 5 steps of the glycolysis. That is, glucose is enzymatically phosphorylated by ATP to yield of fructose 1,6 bisphosphate. This fructose 1,6 bisphosphate, in the presence of enzyme aldolase, that it yield that is, it split of 2 molecules of 3-carbon. That is 2 molecules of 3-carbon products. That is, one is glyceraldehyde 3-phosphate, and another one is dihydroxyacetone phosphate. So up to this, it is called as a preparatory phase. The first phase results is cleavage, that is exose chain. This cleavage is occur 2 ATP molecule. It should activate the glucose to prepare for cleavage into 3 carbon atoms. Then next payoff phase. So the remaining last 5 reactions in the glycolysis is called as a payoff phase. That is, this phase marks it releases the ATP molecules during the conversion of glyceraldehyde 3-phosphate to 2 moles of pyruvate. Okay, listen. This phase, that is the last 5 steps of the reactions, it is called as a payoff phase. Then here the ATP molecules during the conversion glyceraldehyde 3-phosphate to 2 moles of pyruvate. Here 4 moles of the totally in glycolysis pathway 4 moles of ADP phosphorylated to ATP, that is the 4 moles of ATP are formed. The net result 2 moles of ATP per mole of glucose is oxidized, then since 2 moles of ATP are utilized in the phase. So net totally 2 ATP molecules are produced. So first step. So already we told there are 10 enzymes involved, that is the 10 steps are involved in the glycolysis pathway. First 5 it is called as a preparatory phase and the last 5 is called as a payoff phase. Then totally coenzymes, cofactors all are involved in the glycolysis metabolic pathway, already I told. So here we move on to step 1 to step 10. So for what are all the chemical reactions it should be occur in each step, we have to know. So first step, phosphorylation. Here, in the presence of hexokinase enzyme, the glucose is converted to the glucose 6-phosphate. So here, the ATP is converted to the ADP. This is the first step. Then the second step, isomerization. It is a irreversible rearrangement of the chemical structure. That is, the isomerization of the glucose 6-phosphate in the presence of phosphoglucoisomerase, it is converted to the fructose 6-phosphate. Okay, so this is the second step. Then third step is also phosphorylation. Here the fructose 6-phosphate is converted to the fructose 1,6-bisphosphate. Here phosphofructokinase enzyme is PFK. This is a very important enzyme and rate limiting step, that is phosphofructokinase. This is an irreversible reaction and catalyze the phosphofructokinase enzyme. That is a fructose 6-phosphate. It is converted to the fructose 1,6-bisphosphate. Then the step 4. Here the molecules it should be breakdown. That is the 6-carbon sugar. It should be breakdown and converted to the 3-carbon molecules. That is the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by using of enzyme. It is called as a aldolase enzyme. Here, that is the breakdown of molecule. That is the step 4. Then step 5. It is also again the isomerization reaction. It will be occur. Here, the dihydroxyacetone phosphate is oxidized to form the glyceraldehyde 3-phosphate. Then this reaction is catalyzed by the triose phosphate isomerase enzyme. It is involved in that reaction. Here glyceraldehyde 3-phosphate is converted to the dihydroxyacetone phosphate. Okay. Here the 5 steps are involved. Then the step 6 is 2 moles of glyceraldehyde 3-phosphates are oxidized. Here the glyceraldehyde 3-phosphate it is catalyzes to conversion of the 1,3-bisphosphoglycerate. That is the glyceraldehyde 3-phosphoglycerate. It is converted to the 1,3-bisphosphoglycerate. The final resultant reaction, this glyceraldehyde 3-phosphate is converted to the 1,3-bisphosphoglycerate. That is in step 7, the transfer of high energy phosphate group was generated to earlier to ADP to form the ATP. This phosphorylation is the addition of phosphate to ADP to give the ATP. This step is called as substrate level phosphorylation. That is, the phosphate is donor. As the phosphate donor is the substrate 1,3-bisphosphoglycerate. So the end, that is the product of this reaction is 2 molecules of 3-phosphoglycerate. Again, the step 8, that is the 1,3-bisphosphoglycerate is converted to the 3-phosphoglycerate. That is the 2 ADP molecule is converted to the 2 ADP molecule. Here, that is in step 8, the remaining phosphate ester linkages is moved to carbon 3 to carbon 2. Here also breakdown. That is the carbon 3 molecules, it is converted to the carbon 2 molecules. Really in the low energy hydrolysis, that is the 3-phosphoglycerate is converted to the 2-phosphoglycerate. Then 9th step, it is dehydration of 2-phosphoglycerate. Here the enolase enzyme is involved. That is the 2-phosphoglycerate is converted to the phosphoenol pyruvate. One water molecule is going to be removed. Okay. Then it is also a reversible reaction. Then the final step, that is the transfer of phosphate group from phosphoenol pyruvate to adenine diphosphate. That is the reaction is catalyzed by the pyruvate kinase enzyme. That is the conversion of phosphoenol pyruvate to the end product is pyruvate. So totally 10 steps of the reaction, 10 steps of the reactions it should be occur in the glycolysis pathway. The two phases it will be occur, one is payoff phase, preparatory phase, and another one is payoff phase. So totally 10 enzymes are involved. So finally the requirement is 80. Okay. So here how the sum up or how overall the balance sheet of the glycolysis. Here we have to write the glucose, ATP, NAD, H plus and 4 ADP and pyruvate, 2H plus and 4 ATP and water molecule NADH and plus 2 ADP. So cancelling for common terms from these above equation, we get the net equation of glycolysis is like this. That is glucose plus 2 Pi, that is Pi means inorganic phosphate, 2 plus 2 ADP and 2 NAD plus. Then finally overall glycolysis reaction, we are going to be 2 pyruvate molecule, then 2 NADH, 2 ATP and 2 H plus and 2 water molecule. So the stimulation of the reactions involved in the glycolysis are the glucose is oxidized to pyruvate, NAD plus is reduced to NADH and ADP is phosphorylated to ADP. So these three are very important in the glycolysis. Because the glucose is oxidized to pyruvate, then the reduction reaction NAD plus is reduced. That is the reduction reaction, it will be occur converted to NADH and ADP is phosphorylated to give the ATP. So these three reactions are very important in the glycolysis pathway. So this is the energy yield of the diagram. Glucose to glucose 6-phosphate, consumption of ATP 1, and fructose 6-phosphate to fructose 1,6-bisphosphate, consumption of ATP is 1. Then the 1,3-diphosphoglycerate to 3-phosphoglycerate, that is gain of ATP 2 and phosphoenol pyruvate 2. So 2 into 2, 4. Totally 4 gain of ATP. Then consumption of ATP is 2. Then net, what is the gain of ATP? ATP means 4 minus 2. Finally, 2 ATP molecules are going to be yielded in the glycolysis process. So this is the overall reactions, that is the 3 different stages involved, how the glycolysis pathway should be occur. This is the overall reactions involved in. So what is the sum up of the glycolysis? So the sum up of EMP pathway is the partial oxidation of glucose takes place. So the overall reaction in the EMP pathway is glucose plus 2 NAD molecule plus 2 ADP plus 2 inorganic phosphate. It gives 2, the products are 2 pyruvate, 2 NADH, 2 H plus and 2 ATP. So this is the final sum up reaction of the EMP pathway. Okay, it's a very important, you have to write the reactions and structures also, and enzymes and cofactors also you have to write, and finally you have to give the sum up of glycolysis pathway. Definitely you have to write this overall reaction of the glycolysis pathway. Okay, then there are 2 phases occur. Already we discussed. Preparatory phase means the first 5 steps of the EMP pathway, that is known the investment phase. That is the energy is consumed in this process to produce the 2 molecules of trio sugars and phosphates. The payoff phase, the second half of glucose produces ATP and NADH and pyruvate. There are 3 products are given in the payoff phase. That is the second half of glycolysis products are what? ATP, NADH and pyruvate. So location it should be occur in the cytoplasm of living cells. Then ATP it should be utilization and ATP is synthesis and NADH is synthesis. And rate limiting step here is, you just note it down, the rate limiting step. So one word question, one mark question, repeatedly it should be asked in your university exam. So the rate limiting step is phosphofructokinase. The conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by the enzyme phosphofructokinase, PFK. It is a rate limiting step involved in the glycolysis process. So the end product of EMP pathway already we told, 2-pyruvate and ATP and NADH. This is a summary. In your syllabus, that is what are all the significance of the EMP pathway. So we already know what is metabolism, what are all the enzymes involved in the glycolysis pathway, and energy yield diagram, energy yield table. And so finally we move on to what are all the significance involved, what are the significance of EMP pathway. In EMP pathway is called as a universal pathway. EMP pathway is called as a universal pathway of glucose degradation, whether the energy is derived in aerobic respiration or fermentation. In the first step of cellular respiration, and it is required for all tissues to derive energy in the form of ATP, and cells that perform anaerobic respiration derive energy from the process. Cells that lack of mitochondria derive the energy from this process. Krebs cycle and oxidative phosphorylation are occur in mitochondria. So where must the energy is produced during the cellular respiration. So the intermediate formed during the EMP pathway are utilized in the metabolic pathway. In EMP pathway, the various metabolic pathways also occur. That is already I told in introduction class itself, that is HMP shunt, pentose phosphate and forming of triglyceride and fatty acid synthesis, etc. So these are all called as the significance of the embedded layer of Paredes pathway, that is EMP pathway. So here in structure itself we have to write. So we are the here the intermediary metabolism is a major paper, so we cannot write like that. That is the name itself in the pathway. So we have to write the structure. First we have to write how the glucose is converted to the glucose 6-phosphate. So you have to write the structure. Glucose 6-phosphate, glucose structure, then glucose 6-phosphate structure. So in preparatory phase there are totally 5 enzymes are involved. So what are the enzymes? Hexokinase, phosphohexoisomerase, PFK and aldolase and triose phosphate isomerase. So these 5 enzymes are involved in the preparatory phase. So glucose to glucose 6-phosphate and fructose 6-phosphate, fructose 1,6-bisphosphate and glyceraldehyde 3-phosphate plus DHA. Up to this it is called as a preparatory phase. We have to write the structure of this products, reactant and products. So here the structure. So what is the modification it should be occur? We have to easily learn to how we are all well known about what is a glucose structure. Okay, here the glucose structure is there. So in pink color it should be shade, that is the OH hydroxyl group. Here the ATP in the presence of hexokinase enzyme, the ATP is converted to the ATP. What is the cofactor here involved? Mg2 plus is a cofactor involved. So here the glucose 6-phosphate in 6th carbon position, here the phosphate group is added. So this is the glucose structure and this is the glucose 6-phosphate structure. You just note it down. First reaction, phosphorylation reaction, you just draw the structure or write the structure. Then you have to mention what is the enzyme involved in the first step, that is the hexokinase. ATP is converted to ADP and Mg2 plus involved. Then here where the phosphate group is inserted in that first reaction, glucose 6-phosphate. So the second one is isomerization. Here the glucose 6-phosphate is irreversible reaction. Then that is it is glucose 6-phosphate is converted to the fructose 6-phosphate. Here Mg2 plus cofactor is involved. So in glucose 6-phosphate, it is converted to the fructose 6-phosphate, means in first carbon atom and sixth carbon atom we have to add CH2OP. That is CH2OP means what? We have to add both carbon of first position of the carbon and sixth position of the carbon. You have to write add the phosphate. So it is called as a fructose 6-phosphate. So in sixth position alone you have to add the phosphate group. Then the next one is fructose is converted to fructose 1,6 bisphosphate, means you have to add both phosphate group, first carbon atom and sixth carbon atom. Here PFK enzyme is involved. Again the ATP is converted to ADP and Mg2 plus factor is also involved. Then the reaction 4 is cleavage reaction. In cleavage reaction means the fructose 1,6 bisphosphate is converted to, that is the 6 carbon atom, it is converted to the 3 carbon atom. Okay, so you just see this structure. Here 6 carbon atoms are there. Again it is in aldolase enzyme is involved. It is converted to the give the two products. One is dihydroxyacetone and glycerol-D 3-phosphate. Here you see there are 3 carbon atoms are involved. So the 6 carbon atoms, it is splitted into the breakdown into the how much? 3 carbon atoms. So up to this it is called as the first 5 steps involved in the preparatory phase. Okay, then the payoff phase, that is the remaining. So up to this reaction, that is isomerization. Dihydroxyacetone phosphate, it is converted to glycerol-D 3-phosphate. Up to this it is called as a preparatory phase. Then here the overall reaction it should be occur. Then the reaction 6, that is oxidation reaction, it should be occur in that. Here glycerol-D 3-phosphate is converted to the 1,3-bisphosphoglycerate. Here NAD plus is converted to the NADH plus H plus. Here NAD and NADPH are the coenzymes. Here the reduction reaction, that is the oxidation reaction. That is glycerol-D 3-phosphate is converted to the 1,3-bisphosphoglycerate. Here substrate level phosphorylation, reaction 7, that is the 1,3-bisphosphoglycerate is converted to the 3-phosphoglycerate. Then shift of phosphoryl group. Here 3-phosphoglycerate, third carbon atom phosphate group is present. It is shifted into the 2 carbon, second position of the carbon atom. Just see the reaction. Here Mg2 plus cofactor involved and phosphoglycerate mutase enzyme is involved to conversion of 3-phosphoglycerate to 2-phosphoglycerate. Then this 3-phosphoglycerate, again it is converted to the 2-phosphoglycerate. Then the reaction, that is the dehydration reaction, it will be involved. Dehydration means the removal of the water molecule, and enolase enzyme is involved in this step. That is the 2-phosphoglycerate is converted to the phosphoenol pyruvate. So the final reaction, the substrate level phosphorylation, that is the phosphoenol pyruvate is converted to the pyruvate. So this is the overall sum up of the reaction, already we discussed. Here also the overall energy diagram already we discussed. So what is the metabolic phase of the pyruvate means? That is glucose 6-phosphate, it is conversion during the glycolysis process, it is conversion of the pyruvate. So in glycolysis process it is conversion of the pyruvate. So in pyruvate, that is both aerobic and anaerobic process it should be occur. Okay, aerobic means it is the pyruvate it should be formed. In anaerobic means that is a lactate it should be formed. So in fat, the pyruvate in the aerobic condition, the acetyl-CoA it should be formed. Pyruvate in anaerobic, that is lactate, and microorganisms, that is anaerobic microorganisms, the ethanol it should be formed. That during to the fermentation process, the lactic acid it should be formed. So here you see the metabolic fate of pyruvate during the anaerobic conditions. So this final product of the glycolysis, end product of the pyruvate during the anaerobic process, it is absence of oxidation condition. So one important enzyme is involved to conversion of the lactate. So what is this enzyme? That is lactate dehydrogenase. Just LDH, lactate dehydrogenase enzyme is involved, and NADH plus H plus is converted to the NAD plus. So here NADH, the coenzymes are also involved in that. The LDH enzyme, that is the lactate dehydrogenase enzyme, during the anaerobic condition, the pyruvate is converted to the lactate. So what is the fate of pyruvate? So depending upon the availability of oxygen pyruvate, it can enter in any of the following pathways. That is totally two types of fate of pyruvates are involved. That is aerobic respiration and anaerobic respiration. In anaerobic respiration, that is alcoholic or lactic acid formation should be formed. So what is the fate of pyruvate in aerobic respiration? In the presence of oxygen, the pyruvic acid is converted to carbon dioxide and water. Okay, already we know. So the pyruvate is converted to acetyl-CoA, the link of the reactions, and further it is entered into the Krebs cycle. So after the pyruvate is converted to the acetyl-CoA, the acetyl-CoA is the precursor of the TCA cycle. So after the forming of the acetyl-CoA it will be entered into the TCA cycle, or otherwise it is called as a Krebs cycle. So followed by the Krebs cycle, then the electron transport chain and oxidative phosphorylations also occur. So the products formed as a result of this pathway are carbon dioxide and water molecules and energy molecules. Okay, then the products of the resultant pathway are carbon dioxide, water molecule and energy molecules. So this is the fate of pyruvate in aerobic respiration. So then what is the fate of pyruvate in anaerobic respiration? Means in the absence of lack of oxygen, the pyruvate is converted to ethanol and carbon dioxide. Okay, that is, it is called as a alcohol fermentation. In human muscle, under the conditions when there is no sufficient of oxygen available, the lactic acid fermentation it occurs, in which pyruvate is converted to the lactic acid. You just note it down. The fate of pyruvate. Fate of pyruvate depending upon two types. That is the two following pathways. One is aerobic respiration and another one is anaerobic respiration. You just write in aerobic respiration, pyruvic acid is converted to carbon dioxide and water. Pyruvic acid is converted to carbon dioxide and water. The pyruvate is converted to acetyl-CoA. Enter into the TCA cycle, because acetyl-CoA is a precursor of TCA cycle. So what are all the products are formed in the resultant pathway? Means carbon dioxide, water and energy molecules. So that is, the products are formed in this pathway: carbon dioxide, water molecule and energy molecules. Then fate of anaerobic respiration. Fate of pyruvate in anaerobic respiration. The important points are: absence of oxygen, the pyruvate is converted to ethanol and carbon dioxide. That is, it is called as a fermentation process. There is no sufficient of the oxygen is available in the condition in human muscle cell. So the lactic acid fermentation it occurs. It should be occur. The pyruvate is converted to the lactic acid. So that's all about the fate of pyruvate. So already we discussed what is the fate of carbohydrate and what is the fate of pyruvate also. So in glycolysis pathway, or otherwise it is called as a EMP pathway. It is a very important 8-mark question. So you have to write the structure of glycolysis pathway. That is totally 10 enzymes are involved. There are two categories of the phases involved. That is preparatory phase and payoff phase. Then the end products are pyruvate, and in aerobic process it should be give the pyruvate, and in aerobic process it should be give the lactic acid. So in lactic acid, pyruvate is converted to the lactic acid, means in presence of enzyme, that is LDS. It is called as a lactate dehydrogenase enzyme. It is involved. So totally the 10 enzymes, that is the coenzymes and cofactors, all are involved in the glycolysis process. So the net result of the glycolysis metabolism, there are totally 2 ATP molecules it should be required. Okay, so we have to take the carbohydrate in our diet. So it is very important of carbohydrate. Then no skipping of morning breakfast. So it is also very important, because morning skipping of breakfast, it leads to the poor of our metabolic energy. So we have to definitely need to increases our metabolic energy, means we have to take proper food diet and balanced food diet we should take. So here you see in glycolysis pathway, the hexokinase enzyme is involved to conversion of glucose to glucose 6-phosphate, means hexokinase enzyme is involved to conversion of glucose to glucose 6-phosphate. Absence of hexokinase means what it will be happen? Cannot be formed for glucose 6-phosphate. So we need for various types of the diseases also it should be occur. So example, diabetic also it should be occur. Diabetic, so this is diabetic is a hereditary disease, and the insulin is very important for diabetic. There are two types of diabetic: non-insulin dependent diabetic patient and non-insulin dependent diabetic patient. So in the carbohydrate metabolism, so that that is the major metabolism pathways are very important. That is glycolysis, and next we are going to discuss about the TCA cycle, then HMP shunt. Then so these are all the very important pathways in the carbohydrate metabolism. Okay, and that is through to the food only we have to take for the balanced diet. So some of the chemicals, compounds, it cannot be synthesized in our body. So through to the diet food only we have to take to proper the metabolic pathway. We know the liver is a major organ, so all our metabolic pathway should be occur in the liver. So we have to know, we have to monitor the glucose level in our body, because this glucose level variations it should be occur, means it is a fasting blood glucose level and postprandial blood glucose level it should be occur, means in fasting level the normal abnormal condition it should be occur, means it leads to the diabetic mellitus, and we have to monitor the glycosylated hemoglobin, that is HbA1c is very important to know for diabetic profile. It is fast blood sugar level and postprandial blood sugar level, and the final 3 months average blood sugar level we have to know, that is the HbA1c, that is glycosylated hemoglobin level. So the metabolism of glycolysis pathway is very important. Whether the glucose is converted to glucose 6-phosphate, the absence of hexokinase, the product it cannot be formed, means it leads to the diseases. So not only for this metabolic pathway, in all the metabolic pathway also the enzymes are very important, and cofactors also very important. Okay, so that's all about glycolysis.